Friends, today we are going to discuss the cell cycle. Here we see the intact cell. So intact cell in this interphase, that is in G1 phase, it synthesizes RNA and cyclin proteins. All those things are required for the synthesis. So here in the S phase, the DNA replication continues. So before DNA replication to start, the E2F factor need to get released from the RB protein, which is an inhibitor of E2F. So it is done by the phosphorylation. So then it moves to the G2 phase and then M phase. So during you see the a cell, which is a organism divided into eukaryotic and prokaryotic. So the cell which is intact, here you see the intact cell nucleus and DNA within the nuclear membrane. So in the first phase, the nuclear membrane get disappear by disintegrating the cytoskeletal protein laminin A, B, C, which is present in the nuclear membrane. So it disintegrate and disappear. Then you see the the DNA molecules. Now it change after synthesis, after the replication process. Now you see the homologous pair and the sister chromatids, and in this state the chromosome in 4C state, and it with the help of the centrioles, they form the microtubule chromosomal fiber. So it attached to the kinetochore region, and it pulled apart to the poles, and it separate out, and cleavage is gone, and new cell formation is takes place. And this is the structure of the microtubule. You see there are alpha tubule and beta tubule, which think 13 protofilament, and it's 210 to 250 angstrom in diameter. So it is the microtubules. Now you see the regulation of this part during the cell cycle. You will see some checkpoints: G restriction point, G1S, G1 and S checkpoint, G2 M checkpoint, and APC checkpoint in the anaphase between the transition from M phase to A phase, anaphase phase. So in this case particularly what happened, there is a regulation, because in the DNA to DNA replication and then mRNA form, during this process there are several chances that your DNA or the nucleotides get damaged. So if damage is occurred, so need to be stop in the checkpoint. So this work is done by the activation of E1 protein and inactivation of CDC2 and their regulator, because CDC2 helps to activate the CDC that is cyclin B protein, because there you see there is threonine 14 and 15 which get phosphorylated onset of mitosis by dephosphorylation of threonine 15 and 14 by CDC 25. So it is very important. So these are the checkpoints, and this is a cycle you can see. Thank you. The whole video you can. Here you see the cyclin and CDK. CDK is constant, but cyclin is changes progressively G1, S, G2 and M2. Thank you.